today's section. Today we are discussing about scanning electron microscope. The scanning electron microscope is one of the versatile instruments available for the examination and analysis of microstructure morphology and chemical composition characterization. So today, the scanning electron microscope, we are discussing the electron microscope. So in electron microscope, we are using highly energetic electrons to magnify the object or the sample. So by using this electron microscope, we get the information about the sample, its topography, morphology and composition, and crystallographical information about the sample. For using a sample, we get these informations by using electron microscope. So the electron microscopes are classified into two. One is transmission electron microscope and the second one is scanning electron microscope. So transmission electron microscopy is a study of inner structure. So by using this transmission electron microscope, we get the inner structure of that object. And using the scanning electron microscope, we get the information about the surface of that object. So the scanning electron microscopy is visualize the surface of that object. So we are discussing the scanning electron microscopy in detail. So scanning electron microscope. The electron microscope which used a focused beam of electrons to magnify that object, or we get the surface information of that object. So to understand the working of that scanning electron microscope, look at this figure. In this figure, this is the electron beam and here the sample. We are so familiar with these two words, electron beam and sample. And when the electron beam is strike the sample, a backscattered electron and secondary electron and a characteristic X-rays are ejected from this sample. Three things. One is backscattered electron, secondary electrons and the characteristics X-ray. These backscattered electrons are scattered due to the elastic scattering and the secondary electrons are ejected from the surface of the sample and the X-ray is produced or ejected due to the interaction of this sample. So these three are these backscattered electrons, secondary electrons and the characteristics X-rays gives the information of our surface of our object. So by using these three, we analyze these three informations and we get the information about the sample. So next we discuss the basic compounds in scanning electron microscopy. The first one is electron gun. Second one is condenser lens, objective lens and scan coil and detectors. This is a schematic diagram of SEM. The SEM is scanning electron microscope. So first one is the electron gun. The electron gun has produced the electron beam. The electron beam is accelerated towards the downward direction with the help of anode. That means anode is positive potential. And this electron beam is passed through this hole. And we are using a magnetic lens. The next one is the magnetic lens. This magnetic lens is also known as the condenser lens. This condenser lens is focus this electron beam to a small region. The size is approximately 40 to 50 angstrom. So this magnetic lens is also known as the condenser lens to focus that electron beam. Next one is scanning coil. This scanning coil is deflect the electron beam into a particular direction. And we are using another magnetic lens as this. These magnetic lenses are known as objective lens. This objective lens is focus this electron beam to the sample. And we know that this is the specimen. The electron beam is strike this specimen, we get the secondary electron and backscattered electrons. By using a detector, by using a secondary electron detector, also backscattered electron detector, we detect these two electrons. Also, there will be an X-ray. So, these three secondary electrons and backscattered electrons and one is X-ray. These three gives information about our object, surface of our specimen. And this is also a schematic diagram of our scanning electron microscope. And this is electron source, anode, condenser lens, scan coils and objective lens, and the last one is a sample. This is a scanned image of the sample. We get the surface of this sample. Advantages of scanning electron microscope. First one is the resolution of the order of a few nanometer. Second one is information about the elements and the compounds in the sample and their relative abundance. Third one is the biological specimen like pollen grains can be studied. And the disadvantages are that can produce the image of the surface only a few nanometer deep. So next one is the applications. First application is image morphology of the sample. Second one is image compositional and some bonding differences, and examine wet and dry samples while viewing them. And the last one is we can view the frozen material. And last is generate X-ray from the sample for microanalysis. Thank you.